about the applications for DNA. So we talked a lot about DNA, and as we said, it's the blue fingerprint. Okay. So, do you have any idea about like some applications that could be used by using DNA, by using the information of DNA? Basically, like 4 general applications for DNA. So that is related to human. So we have what is called DNA profiling or DNA fingerprinting, which is a technique that they use to analyze the DNAs for a person. So let's say in murder, so we can find out how DNA fingerprints can be used to catch a killer. How they do the investigation thing and find out who's the killer, who's the, when they have like 1, 2, 3 suspects, and so they use the DNA to find out who's the killer. And about the family, did you hear about the kinship?
Yeah.
So what do you mean by kinship?
It's like a connection.
Yes, so it is the blood relationship. So let's say here's the mom, here's the dad, this like his son or their son, or okay, so it's about the blood relation. Let's talk about the murder part. There's an article or about an event that happened like a long time ago in 1988. So this is an article talking about in 1987, 2 teenagers, one of them called Jones and the other called Reish. So we could read a little bit about this event. If anyone could read it for us aloud. Daoud, could you please read it for us?
In the following morning, the fishermen found the bodies of Perry and Brock in the woods adjacent to the recreation area.
This is now the part of applying like DNA information. So, laboratory that specializes in DNA fingerprint testing extracted the DNA from semen sample. That was the evidence that they have. So this is the DNA for the evidence that they found. And here are the blood samples for both of the 2 boys. This is for the suspect 1, which is Reish, and the other one is Jones. So as you can see, if you can look at this, the evidence that they have, and look at the DNA for the 2 suspects. Which one do you like? So from this analysis, they figured out that suspect 2, who's Jones, is the killer. Okay, so they have the evidence from the semen sample, and they have the evidence from both of them blood sample. And then when they did the analysis for the DNA, they found that. And you can compare, so you can see this part, the same like the evidence that they have. To compare the semen sample, the DNA fragment, which is the evidence, this part, with the others, we could, let's say, drag, this is more clear, right? So we could drag this part and slide it and overlap it. Is it for suspect 1? Suspect 2. So that's how they found the killer, by analyzing their DNA from different samples. What I want you to do is, for this worksheet, there's 2 parts. Part 1 is about DNA fingerprinting, and the second part we're going to talk about it in a minute. But for the first part, after we talk about the DNA, analyzing DNA fingerprints, in pairs, I want you to work on first part. It is 2 questions about what we just took, okay? So you can discuss it with your partner. So what are the 4 applications of DNA that could be used? talked about the murder things, if you could explain like how DNA was used to solve the crime. Okay? Brief. Okay?
From there, they were able to ask the murderer himself which was the other's jurors.
This one's in.
Yeah.
Exactly.
Miss, what's the process of the DNA? They call it
You're gonna look into psychology, okay? You will have a post called DNA extraction virtual lab, okay? So we're gonna talk about the DNA extraction lab briefly, and then each one of you will get the chance to do the lab virtually, okay, by your own, step by step. So, could you please log into Schoology? DNA extraction is one of the most modern of the biological sciences. So, it is used to diagnose many medical conditions. Do you remember some of the DNA applications like last time we talked about? Yes, that would.
Like innocence and profiling.
Yes. So it could be used for investigations, for what? For yes, for forensics, and what else? We talked about kinship. So the blood relationship. DNA is extracted from human cells for a variety of reasons. So you will use a pure sample of DNA that can be tested for many things. Okay. So this is the virtual lab. The DNA will be extracted from a cheek swab, okay, from the human cell. Individually, you're gonna start the lab. Yeah. So in this experiment, what you need is to isolate the DNA from a human test subject. Work on DNA extraction. So those are the 4 steps for the DNA extraction. You're gonna start step by step. So individually, work on your experiment. Okay, step by step. Read the instruction. Read the steps. So those are the materials and the equipment that you will use for the to purify DNA from cheek cells. This is the warm water bath, centrifuge, micropipette, the swab over inside of the mouth and collect the sample. Cheek cells has what? Your what? Your DNA. We're putting what? Lysis solution. Okay, so you're gonna think about each step of DNA extraction, why you're using this, why we're doing this. So we use lysis solution. Let's keep it in your mind and think about why we're using lysis solution. Lysis is a Greek word, it means what? Separate. So keep this in your mind to help you figure out why we're using lysis solution. After you're done with the virtual lab, so we're gonna watch a video for the DNA extraction from cheek cells, peeling the fruit map. After you watch the video, okay? So you will be in groups. So taking notes for each step for the DNA extraction. Keep in your mind like why we're doing this, why we're using this solution, okay? What some applications that we could use it for from the DNA extraction. So it's a less than 3 minutes video. So we'll watch it, keep in your mind, taking notes, and then we'll do the peeling the fruit. can do it at home. You can have all the materials. It's not like expensive. You can powder it at your home. And the procedure, as you can see, it is simple. What you only need to do is what? Get what? Yeah. So, do you have any idea about the peeling the fruit map? Peeling the fruit map. You can see here, so we have the map. It's called understanding map. So, the first layer, which is the skin, so you could have your what applications of DNA extraction, what uses for DNA extraction, and then this part under the skin will be the mysterious thing. Like you can write down questions, your questions, why we're doing this, why are we using light solution, what is the purpose of DNA extraction. Okay, so this part under the skin here will be the questions, your questions. And then in the substance part, you're gonna like write down your explanation, your make connections between using some procedures that we did and with the DNA extraction. And then we go deeper and we have the core. The core will be your conclusion. Okay? And connecting it with some applications like blood typing, some diseases that is related to genes. Okay? So in groups, yes, you will be in group size 3 or 4, and then after you're discussing and brainstorming ideas, you will have 4 questions to be answered, okay, before the end of the class, individually, okay? So this part will be a group work, discussing, brainstorming ideas, and then you have 4 questions to be answered. Clear?
Yes.
Clear? Yes. So in the first layer, you're going to talk about what did you see in the DNA extraction lab, what did you notice?
Guys, I'll write our names.
And then write down some questions. Why did you use that? What the purpose of DNA extraction or whatever. And then in the substance part, what you're gonna write is you're gonna explain the whole DNA extraction lab. Okay? Each step, why we're using that, and then you're gonna write here the conclusion. Okay, yes. Write your name. Write your name. Okay. If each one of you has different viewpoints, you can write all your viewpoints, all your ideas. Okay. So, what did you notice from the DNA?
I noticed that it was red. Also, there was the cell was green.
You could also extractors.
Great. The cell first.
Interrupts the cell membrane and the cytoplasm. So then it allows the chromosomes, DNA subunits
Great.
It pulls apart the histone.
Great. So, yes, write all of these ideas in this part, and then you move on, you move into this part. What question do you have?
what was that thing that we used? I think that made it spin.
A centrifuge. So, all of you can
What?
you could add your, what did you notice from the video?
No. Okay, I used to
Yeah.
I write it like this. I used to write it. Why does the salt solution break down?
Very long time.
Solution break down the cell membrane.
That's the whole regulation thing we've been learning. No,
Right,
No, like if the composition of a disease itself is for explaining
Baby cows drink milk.
Disrupts the cell membrane and nuclear envelope.
solution
You didn't specify what.
which
Oh, we're going this way.
Detergent disrupts the
Actually,
cell membrane and nuclear
she didn't say if it was a cow.
detergent
What question? That's my Facebook.
Male or female cows.
membrane
So
and
then add
nuclear
lysine,
envelope.
which can what?
This is the answer to this one.
Mrs. Licker.
it has proteinase K, detergent bursts the cell membrane, and the proteinase K unwraps the histones.
What is DNA composed of?
Genetic genes.
Okay.
Because it has detergent.
Deoxyribonucleic
I know.
Blood sugars.
Because it has detergent.
Or nucleotide sugars.
proteinase
Nucleotides, proteins, and sugars.
Okay, and it like it breaks it down or something.
Yeah.
Do you believe in aliens?
Develop like.
Do you believe that aliens are
So
real?
their DNA, if they
Because
have
I
it,
do.
they would include it in those to the point of like
Yeah.
unrecognizable.
Yeah, you're
We're friends again. Wait, what did you say?
They're an alien, of course, I
We
forgot.
will have a deep discussion together.
To log into your Schoology, there are 4 questions to answer. Okay? Related to the DNA extraction doubt. Okay?
Which exercise is this? Or so we'll figure out what it is.
How traits pass through DNA. Okay, so we're talking about now traits, which biomolecule that do much of the chemical work inside the cell. The traits.
The traits of a living thing depend on the complex mixture of interaction components inside it. Proteins do much of the chemical work inside cells, so they largely determine what those traits are, but those proteins owe their existence to the DNA.
So to understand how this all comes together, consider the trait for blue eyes. So thinking about like you have blue eyes, you have brown hair, you have freckles. So DNA for your blue eyes gene is copied as blue eyes RNA message. Okay, blue eyes gene is copied as blue eyes RNA message. And then that message is translated into blue protein pigment found in the cells of the eye. So for every trait we have, like your eye color, your skin color, or your blood type, or your hair color, there is a gene or a group of genes that controls the trait. By producing what? By producing first the message, which is the RNA, and then the protein. So what I'm going to do is to read this article, okay? And we're gonna do the what is called corner notes. You can use your Google Doc. Yes. Yes. Corner notes. You're gonna have 2 columns. First column gonna be like the main points. It could be a questions or let's say the main points, okay? And the second column will be like in details. You have 40 minutes, so you have short 8 paragraphs. So you can have 1 point for each paragraph, okay? about is in the last 2 minutes of the genetics in Harry Potter world. So, from last class, like about the basic genetic terms that you learned about phenotypes, genotypes, dominant, recessive, and so we're gonna talk about like what types of inherited genetic traits are described in Harry Potter series. All the weasels have red hair, freckles, and more children than they can afford. The second one is he was almost twice as tall as normal man and at least 5 times as wide. Harry had a thin face, no knees, black hair, bright green eyes. What about the last one? A pale boy with a pointed face with white blonde hair. Do you remember who's the magic kid?
The mean kid.
Me, Draco.
Okay. So, can you tell me what are the 4 traits we're talking about? And the first one is what? We're talking about red head, freckles, and the 2 possible phenotypes for freckles are someone could have freckles and someone couldn't have freckles. So no freckles and has freckles. And both are observable, right? So you can see if someone has freckles, you can see them.